So we continue with the hippocampus today. Before I continue, I wanted two notes. I don't know if you got the email from Moodle, but I've placed the schedule for presentations on Moodle. Even though it doesn't look like that, there are way more students that take the course for credit, so we have like 4 more presentations than I planned. So I had to add 2 sessions. One of them will be actually in a week and a half. One class before we planned, and I will send these 2 people the papers for the presentation today. And the other thing I want to say, if anyone is downloading the presentation, there are new slides on the one from last week, from Tuesday. So you might want to re-download it. I uploaded the updated one. Okay, so we were talking about LTP. So today we're going to continue with LTP. A little bit on the mechanistic side, and then we're going to switch to more functional aspects of the hippocampus. There are so many things to say about it that it's just impossible to cover them all. So I chose a few of them, and I skipped at least 2 that I thought were very interesting, but there wouldn't be time to cover. So we might cover them with a paper. Okay, so here again is the phenomena, a cartoon of the phenomena of LTP. You have say a CD1 pyramidal cell, for example. You have 2 pathways. One is a control, one is a tetanized one. You test them, and you get on the control pathway you get an EPSP. Every time you give a stimulus, you get an EPSP. On the test pathway, every time you give it a stimulus, you get an EPSP. There's obviously some variability. Then you give a tetanus only to the test pathway. And this is what it would look like because you stimulate it many times, so there's a big summation of EPSPs. And after that, when you start testing, what you get is like larger EPSPs on the tetanized side, and no change on average on the control side. So this is the basic phenomena of LTP, similar to what we were seeing on the Bliss and Lomo paper for 1973. And now the question, what's going on? So obviously this looks like an important phenomena because we know that the brain learns, that there's plasticity, and this looks like a really good candidate for plasticity. It has some Hebbian aspects, as we say. It has some relation to the input and the output. But what is going on? How does that happen? And this was the target for infinite amount of studies, and it's still not figured out completely. We know really a lot about it, but it's not. There are still many open questions, and I'll try to hint to them. So some of the things that kind of we know, you know as well. Again, so this is a phenomena. You plot the amplitude of each one of the EPSPs, and after a while, after the tetanus, you have a change in the average amplitude, and this lasts for as long as you can measure. Okay. Okay. So one thing that we know, which is a key, is the fact that the EPSP is mediated by 2 types of receptors. One of them is the AMPAR receptor, if you've never heard of that, let me know, okay? Because I assume that you know what an AMPA and NMDA receptors are, but just briefly, So AMPA is a receptor for glutamate. Glutamate binds to the receptor, and then sodium and potassium get in and out. The permeability to sodium and potassium is changed, but there's more sodium coming in than potassium out, and the net flow is actually a positive charge into the inside of the cell, and the cell is depolarizing. The other receptor is also a receptor for glutamate. It has two binding sites for glutamate, but in order for it to open, there is this site of magnesium block, and then when magnesium is in the medium, it blocks the receptor in a voltage-dependent manner. So when magnesium, when the membrane potential, the difference between the potential inside and outside is very hyperpolarized, then the magnesium would block the receptor, and if it depolarized, the probability of the magnesium to leave is higher, and then the receptor also become a channel for ions to flow in, and in this case, there's a flow of also of calcium through this receptor. So this drug, which is called AP5, and the 5 is Roman, so sometimes it's called APV, is a drug that blocks NMDA completely, independent of the membrane potential. Okay. And then you can see that if you hold the cell at if the cell you hold the cell, it means that you have like an electrode in the cell, and you inject current such that the membrane potential would be -70, even if the resting membrane potential, say it's -50, you can still hold it at -70. But you know, the holding is a tricky word, because usually when you say you hold the cell at -70, it means you do voltage clamp. So you kind of really hold the cell at -70, and then nothing changes the membrane potential. The case here is current clamp, because what we measure is voltage. Okay, but we still can shift the membrane potential. So the average would be -70. And then if we activate the synapse, we get an EPSP, and if we add AP5, basically nothing happens. Okay, it doesn't happen because at -70 the NMDA receptors are blocked by magnesium, and then if you block the NMDA receptor on top of that with AP5, nothing will happen. However, if we depolarize the membrane to -40, then there's a big difference if you add AP5, and basically the difference between that curve and that curve is the contribution of the current that goes through the NMDA receptor. Okay. You can do the same trick in voltage clamp. So you hold the cell at -95, and you get a current, and this is the current through at -95. The current doesn't change a lot. Again, because when you apply AP5, the NMDA receptors are blocked anyway. Okay, but if you hold it depolarized, then when you apply AP5, you block a lot of the current that's flowing through the. So in this case, at 30 millivolts, most of the current is NMDA. And note that the current direction has changed. Okay, you see, this is the current that goes inside because we hold the cell hyperpolarized, and this is the current that goes outside. What does that tell you? Anyone kind of, is it like Chinese, or everybody understand what we're talking about? No? Sorry?
Last slide.
Previous slide?
Untitled.
Okay, so you have a synapse, and you have an action potential comes here, and glutamate is released from the synapse. Okay, from the presynaptic areas. If glutamate goes and binds AMPA receptor, and also binds NMDA receptors. If the membrane potential is minus 70, never use animation. If the membrane potential is minus 70, then this magnesium is blocking the channel. Nothing goes through this channel. So everything that goes through the channel, everything that goes in would only go through this channel. Okay, and then if you will add a blocker that will block that channel, nothing will happen. Okay. So this is this case. So you add a blocker, nothing happens.
The green one?
It's one of them is a green, and then you add the drug, and you get the yellow one, and nothing changes. It's the same thing. Okay. Now you do the same thing again, but now you hold the cell at minus 40 millivolts. At 40 millivolts, these channels, the magnesium block is released, so the magnesium just goes away. So the channel now can be opened if glutamate will bind to it. Okay, glutamate binds to the channel. Now current goes through both channels. Okay, since you have now more channels that open, you get more current, you'll get more voltage. This is what happens in the green one. Now you add a blocker, which is not the magnesium blocker, just another thing that blocks it. And then you get this current, this voltage. And the difference between them is actually due to the fact that in minus 40 you have additional channels that are open. Okay? But this one is a little bit different. It's the same story, but channels, these ion channels or these receptors, have what we call the reversal potential. It's due to the selectivity to how much potassium relative to sodium is getting in and out. Okay, and if you go to plus 30 millivolts, you are beyond the reversal potential. So now when glutamates get in, actually more potassium goes out than sodium gets in. Okay, and then what happens is that the current actually goes out. You get a reversal in the direction of the current. So somewhere in between minus 95 and plus 30, the synapse is reversing the direction of the current. Okay, and we find that by actually plotting the voltage, the change in the voltage, as a function of the current. The change in the current as a function of the holding voltage. Okay, and in this case, you see that the reversal potential is about 0 millivolt, and this is actually true for both NMDA, almost both NMDA and AMPA receptor. Okay, but now we can do the same thing here. We can do the same thing again, but now instead of blocking the NMDA, we'll block the AMPA receptor. So we'll be left only with NMDA current. Okay, so at minus 75, in control, where we have both, we get a current. Okay, but if we block the AMPA current, we get almost nothing from the NMDA. Okay, if we go now to plus 50, in control, again, the current is reversed, you see. But if we block the AMPA receptor, we still have a huge current, which is almost the same. Okay? So what happens is that if you plot now the curve in the presence of CaMKII, so everything you have is only NMDA, you get this kind of curve. Okay, this curve. And this curve is a non-linear, voltage current relationship. Okay, the current is dependent on the voltage in a non-linear way. So here it was linear. Where was it? Here. This is linear. Okay. This is non-linear. Okay, it depends on the voltage, and what happens is that at some voltages, around -40, you get the maximum conductance, the maximum inward current, and then if you go and depolarize the membrane more and more, you're starting to get outward current. Okay, and the reversal is also very close to zero. So this is a reversal potential. Now I told you that one of the reasons for that thing, okay, that at -80 you don't get any NMDA current is because of the magnesium block. So what happens if we don't have any magnesium in the medium? If we don't have any magnesium in the medium, there's nothing to block the NMDA current, and this is what you see here. So it's like a linearized version of the NMDA voltage current relationship. Okay, so this is zero magnesium. So now you do like a tetanus in a way, okay, in control, and with APV, and you see that there is actually a big difference in the response with APV and with control. Okay, so when you see that, what it basically tells you is that when you give a single EPSP, and the cell is at resting membrane potential around -60 millivolt, every EPSP is mainly due to AMPAR receptors. Okay, there's no NMDA. But when you do the tetanus, you actually depolarize the membrane high enough to start getting the NMDA unblocked. And then when the NMDA is unblocked, it adds some current, so you get a bigger EPSP. What is important for us is actually, okay, what is important is actually that some of the current that's responsible for this thing is what? It's carried by what? By calcium ions. And calcium, as you may know, is like the key to everything. When calcium gets into the cell, things start to happen, right? And so this was the first clue for starting to look for the mechanisms of how these things could work. Okay, so people started to really work hard on this NMDA receptor. First of all, to see if indeed this idea actually holds water, if this is actually the mechanism in which by which LTP is working. And I can tell you, as an anecdote, I haven't put it here, but more or less together with the discovery of LTP in the hippocampus, there was a discovery of LTD in the cerebellum. And the cerebellum is so David Marr, who had a model of how the hippocampus should work, also had a very influential model on the cerebellum, and there he suggested that the main trick would be also LTD. And indeed, there was a famous scientist in Japan called Ito, who did similar experiment by doing tetanus activation of Purkinje cells and found a long-term depression. So synapses, instead of actually being potentiated, became depressed, smaller. And interestingly enough, in the cerebellum, in the Purkinje cells, there are no NMDA receptors. Okay, we know that today. They didn't know that at the time, but there aren't any NMDA receptors. So it's a different mechanism altogether. Okay, so NMDA receptor, I'm not going to go much into the details, but basically it's a complicated protein. It's built from 4 subunits, and some of these subunits can be exchanged. So you have all kinds of variants of the same NMDA, not of the same NMDA. You have different variants of NMDA receptors. One of the interesting things that happens is at some point during development, this subunit, which is NR2B, is replaced by NR2A, and actually this happens. So NMDA receptors, it's another story, but there's a lot of arguments about it, but it is apparently present in postsynaptic sites, but also on presynaptic sites. And there are period where the NMDA receptor on the postsynaptic side is switched from NR2B to NR2A, but on the presynaptic side is not. And then you can have selective drugs that will block the postsynaptic and not the presynaptic, all kinds of tricks like that. What else did I want? But you could, people have shown that at least in some cases, like in CA1 pyramidal, Schaefer collaterals, CA1 pyramidal neurons, LTP, NMDA receptor is critical to get the LTP. So if you, for example, knock out the two indications. First, if you apply AP5 during the tetanus, so you have LTP. So it's, again, protocol. You check the amplitude of the synapse again and again. Then you do a tetanus. You get LTP, and then it stays. The amplitude of the EPSP stays high. Okay. Now you repeat the experiment, of course, on a different preparation. And during the tetanus, you apply AP5, but only only during the tetanus. Okay, and what you get is actually you don't get LTP. It goes back to baseline, right? So during the induction of LTP, you need the NMDA receptor. Now, if you repeat the same thing, but you apply it later on, okay, nothing changes. So once LTP has started, you don't need the NMDA receptor to continue to carry it. Okay, so something like the obvious conclusion, which is true or not true, this is debatable, but something is changed in the AMPAR receptors, but not in the NMDA receptors. Okay, so because when you do that, it doesn't actually say that nothing happens to the NMDA receptor, but it says that during the testing here, the membrane potential is low and the amplitude of the EPSP is not so big, so you're not going to engage NMDA receptors anyway. Okay, so this is what you get here. If you knock out one of the subunits of the NMDA receptor, then you don't get LTP. Okay, and interestingly enough, if you can knock out the NMDA receptors only in CA1 pyramidal neurons, and then you still get LTP in the dentate gyrus. Okay, so in these synapses you'll have NMDA, you can get LTP, you don't get it in CA1. Yes?
In the top picture, what is the APV administered?
So here it is administered in the green one, just somewhere here during the testing, after the induction of LTP. Okay. So, yes.
I just wanted to ask about the top units. Are they different genes, or is it alternative splicing or something?
I think there are different genes, but I'm not sure. It could be alternative splicing. We can check that later. Okay, so now there's a big issue. I for some reason. Okay, so this is a little bit mixed up. So I'll tell you what I wanted to tell you here, but we'll go back to that later again.
So when LTP happens, what causes the potentiation? Does it increase the normal receptors?
We'll talk about that in a second. I mean, I'm not going to talk about all of the possibilities, but yeah. One of the things that okay, I'll go back to this slide again in a second. You remember that last time when we talked about CA1 pyramidal neurons, we said that when you have a spike in the soma, the spike back propagates into the dendrites. Okay, so in fact, I can tell you a personal story here. When I came first, and I was kind of looking for PhD advisor, and I was talking to Idan Segev. I came to him, and I was asking him exactly this question. Basically, how does the synapse know when you do the tetanus? How does the synapse know? It's like Hebbian and everything. how does the synapse know that there was a spike in the soma when you do this tetanus, right? Because like according to classical Hebbian plasticity, it's like fire together, wire together, right? So you need a presynaptic spike and a postsynaptic spike in order to change the synapse. But the synapse is somewhere in the dendrite. It doesn't know what happens in the soma. How do you actually explain this plasticity? And he gave me this kind of explanation, right? So he said, where was it? Yeah, this thing, right? We have like many synapses acting together. They will depolarize the dendrite. They will activate NMDA receptors. You'll have what you want. But this is kind of very partial because this could happen with the postsynaptic neuron firing or not. Okay, so it's not classical Hebbian in the sense that you need them both to spike. And he gave me a bunch of papers to read, and when I came back, it was like a few weeks later, there was this paper that actually described this backpropagating action potential into the dendrite, and suddenly there was like a much better explanation. So indeed, when this was kind of found, people started to realize that it could be something more to do with these backpropagating action potentials, and they started to think, okay, so now we have a presynaptic spike, presynaptic action potential that activates the synapse, and now let's see what happens if the spike in the soma, we can have like an electrode in the soma of the postsynaptic neuron, and try to control the timing of the spike. What will happen if we make the spike happen before we stimulate the synapse or after stimulating the synapse? And what they found is this quite sophisticated phenomena which is called spike timing dependent plasticity. And the idea is that if you have the presynaptic spike coming before after the postsynaptic spike, so the postsynaptic spike coming before you activate the synapse, you actually get reduction in the efficacy of the synapse. So the protocol is the following: you have say in the hippocampus, all right. You have in this case what was done is paired recording with two neurons in a culture. So they take. You remember that I told you that hippocampus is very good because you can take these neurons where they're young, put them in a dish, and actually grow them as a culture, and then you have very good access to every one of the neurons. So this was done by a guy called B when he was in a lab in Berkeley of someone called Mu-Ming Pu, who is a very influential guy, and they were recording from pairs of neurons more or less at the same time, or even before. There was the same, actually before, by Henry Markram, did the same thing in cortex when he was in Bert Sackman's lab. And even before that, there was a guy called De Ban who was doing that in CA1 pyramidal neurons, but not with pairs of neurons, just by activating the fibers. So the protocol is the following: you activate a single synapse and you record from the soma, and you have a baseline of what the synapse amplitude, what the amplitude of the EPSP. Okay. Now you activate the synapse and activate the postsynaptic cell, say 10 milliseconds before you activate the synapse. Okay, you do that like 10 times or 20 times, and then again you activate only the synapse and check what the amplitude of the EPSP. If you activated the neuron, let's say 80 milliseconds before you activated the synapse, there's no change in the amplitude, nothing happened. If you activate it 10 milliseconds before you activate the synapse decreases in like 25%. It becomes smaller. Okay. If you activate it 10 milliseconds after this, then the synapse becomes bigger, 50% bigger. All right. So actually, the precise timing of the presynaptic and the postsynaptic neurons are extremely important, and this has been found since then almost anywhere you want to look for it. It's in the cerebellum, it's in the cortex, it's in electric fish. Everywhere you look for it, you'll find it. And it kind of was very tricky to interpret. There was like a storm of theoretical papers about what does that mean and what does it mean to spike timing, because if we believe that timing of spikes is kind of random, then what is that, and it would kind of average out and all the synapses will never change. There's a lot of issue about this phenomena. If you want, we can discuss it at some point, but I just wanted to mention it here. There's also a lot of work about the dendritic mechanisms that enable that. We'll skip that now, for now. So,
just short question from the figure. It looks like that the increase can be higher than the decrease.
Right.
The increase phenomenon is in general stronger. Is it just in this figure, or is it in general?
So again, as you see, it's very noisy. So there's like people are fitting these two exponentials, and then people started to, from theoretical point of view, you have to have the integrals of both to be the same. Otherwise, you'll have all the synapses potentiating all the time or something like that. And indeed, in some cases, like in layer 2-3 in cortex, you can show that the LTD window is longer. It's smaller, but it's longer. And it's, so when people were doing modeling of that, they were kind of normalizing the sizes, so it would kind of be 0. But it's kind of not clear what happens really in reality. It's even way more complicated because these guys were kind of taking it to the extreme where they started to look for what will happen if you have like triplets or quadruplets of spikes. So you have presynaptic spike, postsynaptic spike, presynaptic spike. So presynaptic, postsynaptic, postsynaptic, presynaptic. And all this combination, and it becomes very complicated because each one of them. So this is only true for 2 spikes that are completely isolated from everything else. If you add another spike into it, then you can imagine that you have 2 backpropagating action potentials. They kind of summate, activating all kinds of dendritic currents. It's a mess. So this is a very partial view of what's going on there.
So is the backpropagating action potential necessary for this phenomenon? Or?
Yeah. Yes. Exactly. So this has been shown in. It has been shown in '96 that it's necessary for LTP, and then for that it has been shown later on. Yes, I mean you can for this specific one, yes. And also depending on which cell. So for example, in Purkinje cell it's not, because in Purkinje cell there is no backpropagating action potential. So okay, so now there was another thing that is like all the time around LTP is whether this. So here we go back to this picture. Is it? Why is the change? Well, what is the change here in the postsynaptic cell, or is it change in the presynaptic cell, or is it in both? Okay, and this was like there were conferences around this question. Is it postsynaptic or presynaptic? And people were arguing it forever. And if it is presynaptic, how can it actually happen? You have NMDA activation in the postsynaptic cell. What the hell? How would that change anything in the presynaptic cell? It's a completely different cell. He doesn't know about the NMDA activation. There's nothing to do with it, right? So there were all kinds of ideas. For example, that when NMDA opens, there's like a cascade. The calcium gets in, it binds to IP3. All kinds of things happen. At the end, there's a substance called NO that can diffuse through the membrane. Okay, goes to the extracellular medium, and then goes into the presynaptic terminal and start all kinds of processes there. Okay, then there were also, they found that on the presynaptic areas, terminals are receptors for example, for endocannabinoids. So endocannabinoids is another molecule that could be synthesized in the postsynaptic cell, but released to the presynaptic, to the medium, and then take up by the presynaptic site. But also, all these things are kind of very complicated in the sense that, there are tons of other terminals here, right? It's all packed with terminals. So how do you control where this NO is going, where this endocannabinoid is going? When NMDA is like also, so there were also people talking about NMDA released, glutamate is released, but also going back on the same, on these autoreceptors. So the presynaptic cell also have a receptor for NMDA, as I told you before. It's a very complicated question, and recently, so I'm going to show you 2 slides with evidence for changes here and for changes here, and I think today people kind of more or less agree that it certainly happens in both, okay, but there are still lots of complications. One of the things that people are starting to talk about, and not enough, I think, is the fact that these are not just 2 things that are floating around. They're actually molecules that are coming out from here and and kind of hold the synapse together. These molecules are called adhesion molecules, and they hold it. The synapse is actually, this gap here is tiny. Is like 200 angstrom. Is extremely small. And there are actual molecules that connect the 2 things together, and these molecules could actually, apparently, transmit some information from one cell to the other. So when one thing happens on one side of the molecule, it could actually affect the presynaptic side, or the reverse direction. And some of these things we know that happens in. That there are another type of synapse in the body, which is immuno synapse. So when 2 cell and immunocell is contacting a different cell of the body, there's something that the immunologist calls a synapse. And it's called a synapse because they bind together, again, also very close to each other, and start talking to each other, but they're talking with molecules. So there's like molecules coming out, meeting other molecules that are coming from the presynaptic site and talking to each other. So we don't know almost anything about what happens here. It's of course much smaller than in this contact between immunocells and regular cells. But this is an area that is currently very hot. Try to explain, this mechanism in terms of molecular interactions. So 2 things that I wanted to show you about evidence. These are kind of old evidence for postsynaptic and presynaptic. So one of the things that you can see is that, so you have an LTP experiment. You have a change in you have a change in the average EPSP, but the what we call the paired pulse ratio. So if you give a pulse to the axon, and you get an EPSP, and then immediately after that you give another one. So usually the second one is smaller than the first one. Okay, and it depends on the type of the synapse. So sometimes it's smaller and sometimes it's higher, but there is a change in the probability of release of the axon depending on the history of if there was a spike before or after. So people use that as an indication for changes in the presynaptic site. So, for example, you can see here that during the tetanus there is a change in the pulse ratio, but afterwards there is no change. So they take this as an indication that nothing on the presynaptic cell side has changed, but the postsynaptic side has changed. Another indication, which is a little bit more new, is this issue of CaMKII 2. So what you see here is an imaging of a dendrite and a spine, and we know that on the top of the spine there is a synapse. Okay, and this is just a fluorescent dye. Okay, so you see the synapse in both cases. Okay, but here you have a you image CaMKII 2 LTP, so you kind of label the CaMKII 2 with GFP. So you label the CaMKII 2 with GFP, and after the induction of LTP, you see that there's like translocation of CaMKII 2 into the synapse. Okay, and we can people have shown that CaMKII 2 is at least in pyramidal cell, which I think it is expressed only in pyramidal cells, and we use it also as an indication when we want to transfect neurons with viruses to pyramidal cells. So it has been shown that it is involved in the induction of LTP, and here that you have an indication that on the postsynaptic cell there is it actually moves to the synapse that was activated. It also this is an even better demonstration, because what they do here is they do it to two neighboring synapses. So something very similar, but you induce LTP on one spine, but not on another one, and you can see that the CaMKII 2 is getting only onto the synapse that undergoes the LTP, but not to the other one.
Where is the other one?
Yeah, this one I think here. No, this one. So this is the activated one, and this is the inactivated one.
The same part in the lower speaker?
Yeah, I think so. Okay. Presynaptic. So presynaptic, so this is why it becomes very confusing, because, okay, so this is like a very, very type of old demonstration for something happens in the presynaptic cell. So what could happen in the presynaptic cell? Basically, in the presynaptic terminal, two things could happen. One is that the probability of release will increase. So every time you have a spike, you'll have higher probability of release. So on average, so maybe each one of the EPSPs will look the same, but on average they will be bigger, because you'll have more times when you have activation of the synapse. Or if you record like for many synapses like you do in classical LTP, you'll have each time you have more synapses active, so probability of release. The second thing that could happen is this what they call the quantal content. So you can have more glutamate released from a vesicle. Okay, so one way to do one of the first old ways to do that would be to load the preparation with radioactive glutamate, H3, doesn't matter, and then induce the LTP, and kind of you can measure how much glutamate is released before and after. Okay, and this is what you see here, and you see that there is an increase in the release of glutamate. But this is extremely kind of crude measure. You could also do that with a little bit more, higher resolution. Not with that method, but here what you look at is that you look at the failure ratio, and you see that the failure ratio is decreased. So every time you give a spike to the presynaptic site, what's the probability of getting an EPSP? And if this is actually decreasing, sorry, if the failure rate decreases, it means that the probability of getting an EPSP is increasing, and this is translated into a presynaptic mechanism, because a presynaptic mechanism is the postsynaptic mechanism is not shouldn't have any failure ratio. Whenever it has glutamate, it will give you an EPSP. The receptor always open when they have glutamate. It don't have like any probabilistic thing going on there. And whenever you have an EPSP, the size is the same. So in a way, this is kind of This is why there were all these controversies, because this is almost the opposite result of that, right? So sometimes you get, some people got that, and some people got that, and this was depending on which synapse they were using, what they had in their pipette, what was the temperature. It's, impossible. I will show you in a second. Maybe I'll show you first. So when you go in that book that I showed you on the hippocampus, on this chapter of synaptic plasticity, it ends at page 444, where the reference starts, and the reference ends at 474. So 30 pages later, you end up looking for references. Okay, and it all has to do with LTP. It's impossible to kind of figure out that thing. So beware if you choose that as a question. But what I really wanted to highlight is this four open questions regarding that thing. So obviously it's a very important phenomenon, but there are some things that are kind of completely obscure. At least when you look at all these thousands of references, there is no clear answer to some of them. It's clear that protein synthesis is required for LTP. Okay, we talked about that. But protein synthesis happens in the nucleus. The synapse is like, millimeters away. We're not even talking about the presynaptic terminal, that if something has to be presynaptic, it can happen meters away from the nucleus. All right? And then it's meters away from the nucleus, but it could be like micron away from another synapse that didn't go LTP. So this is the huge question, how do you actually do that? And so there is something that has to go from the synapse to the nucleus, tell it that it needs to, we need to do something to synthesize proteins, and then go back to the synapse from the nucleus and actually hit the specific synapse. Okay, so some of these pathways has been kind of found. This CREB is a very important molecule in this process, and all kinds of other kinases, but it's still, it's really, really unclear. Then you have the issue of how to go from the nucleus back to the synapse. And there is a theory which is called synaptic tagging. So the synapse is like kind of raising some tag and making itself recognized by the mRNA that goes back to the dendrites, or by the proteins that are carried back to the dendrites. But again, this tagging is a very unclear thing, because it's not like, you have a complete set of, markers. For each synapse it has a name with a tag, like you have a barcode on the supermarket. It's not clear how this is being so specific to one synapse and not to another one. So it kind of says, in a way, if you believe that, it kind of says, it can say, I have to go LTP, but if I want to go 20%, 40%, 50%, this is much more complicated to mark. Okay? Now, the third question here is, LTP can happen once and hold forever. At least as far as we know, it holds for a very long time. But proteins don't, they don't exist forever. They are being recycled. So say you have a synapse that undergone LTP, and now it has more AMPA receptors in the synapse than it used to have before. How do you actually keep a stable mechanism that keeps having this amount of proteins stay the same all the time? Okay, because there's a process of turnover. So something has to remember in the synapse. It's not the fact that you have more proteins just makes the synapse stronger, but this is not the memory. Something has to be there. Remember that you need more receptors. We don't understand that as well. And lastly is what I talked to you about, the presynaptic side. How do they communicate? So you have something happening on the postsynaptic cell, how does it goes back to the presynaptic side? And even more complicated, if and only, and we don't know about that, and I think there was one paper by this guy, Muming Pu, that I told you before, that shows that there is, but this is a very spooky paper, I don't know if you can trust it, that basically shows that there is an information transfer from the presynaptic terminal back to its nucleus, through the axon, going back in the axon. Okay, this is against the dogma big time, so no one actually believes that, but who knows? Okay, so of course we haven't talked about things like other type of, so there are these, you remember I told you there are these massive synapses which are called multi-fiber synapses on CH3 pyramidal cells. I skipped that. These are interesting synapses for themselves. We haven't talked about neuromodulation. So all these things are, are modulated by all kinds of substances, and also other type of substances called like trophic factors. One of them, very famous, is BDNF, which if you add it, you can get really easily LTP. If you take it away, you might not get it at all, depending on the synapse, depending on the dendrite, depending on many things. We talked about that, and the biggest question is, we're going to talk about memory in a second. With 30, I don't know, it's not 30, it's like 40 years now of since the discovery of LTP, this question is still open. Does LTP has anything to do with memory? And I think it's remarkable. We don't have a very clear example that LTP equals memory. Okay, so this is something to keep in mind. If you want a Nobel Prize, that's a good question to start. Okay. So after the break, we'll talk about some of this stuff, and then we'll finish. Okay, so second lecture, and this will be a little bit more interesting, I hope. So the big question is, what does the hippocampus do, right? Is it a valid question at all? Is it actually relevant in a structure like the brain, taking one piece of the brain and asking what it does? Because it's obviously everything is talking to everything, and everything is working together. And there were this was very, there were, in the history of neuroscience, there were eras where people attributed all kinds of functions to different places in the brain, or to inhomogeneities in your skull, to all kinds of things like that. And obviously the hippocampus didn't escape that, and there were all kinds of functions attributed to it, having to do with emotions, with smell, with all kinds of stuff. The consensus today is that it has something to do with memory, and mainly due to this guy, which now we can actually name fully, but for many years was known by HM, and probably this is how it will be remembered. Anyone not heard of HM before?
5 times at least.
Okay. So yeah, I would be happy to completely skip that. So I only devoted like 3 or 4 slides just to remind you what you already know. The textbook says, this is a brain of this is a normal brain, and this is a brain of HM, and they took the whole hippocampus. Obviously, you cannot take only the hippocampus. They also took some of the amygdala. They also took some of the temporal lobe. Basically, he went under surgery in 1953, and then 1957 started appearing the reports about what happened to him. The surgery was because of epilepsy, and since 1957 he was a mouse, if you want. he basically was just studied all the time, and it's kind of, it's an interesting phenomena, together with the fact that the person cannot remember who you are when you can come into the room, so you can kind of do an experiment after experiment, doesn't even remember that he did an experiment. So it's kind of almost classical, but it's, I don't know, it's ethically a bit kind of weird. This is textbook, this is kind of real life. I don't know if any of you is trained radiologist or something of the sort. It's kind of not that easy to understand exactly what is missing and what's not, and obviously it's not just the hippocampus. And you have to remember, okay, we'll show. Okay, so the basic idea, the basic thing that was shown is that he was suffering from what is called anterograde amnesia. So they kind of divide amnesia to retrograde, which is basically what happened before something and what happened after. So in HM's case, it's the surgery, which happened here. He didn't remember anything happening after the surgery, but he did remember stuff that happened before. And there's arguments about how much he remembered. At the beginning they said 2 years, and as I said, 11 years. He could also remember something that happened after the surgery, so this is not exactly complete. And one thing that you have to remember, that until the surgery, he also suffered from epilepsy, and a very severe epilepsy. So this is also something that is kind of the fact that he didn't remember that, or he didn't remember that, it could also be due to the epilepsy. And also some of the things that he had afterwards could be due to the epilepsy. So some examples of what he could and couldn't do if you give him this test, which you get some picture and then you have to copy it. If he sees the picture, he can completely copy it, so he doesn't have any problem, not with vision, not with motor skills, and not even with what we call the working memory, because when you draw this line, you have to kind of keep something in your mind to do that. If you give it him, like, I don't know, I think half an hour later, he doesn't even remember that he's seen the picture. There's nothing that he can do. Normal people can do some of it. You can test yourself what you'll remember out of this picture, in the next lecture, but he doesn't even remember that he saw it. On the other hand, he had these, for example, if you give him a test which is tracing these lines inside in this star-shaped maze, and he wasn't like this, obviously he wasn't, it's not that he couldn't do that, it's just that what they do is they show you it in a mirror, so you can, your hand is going exactly the opposite direction to what you want, and it takes some time to get used to that. So he was improving with time, and you see that after a few days he could actually do it very quickly and very, very well. So something obviously has changed, and he learned something, but this is nothing, but of course every time he saw the picture, he couldn't remember that he did the test. Okay, so it's something else. So they kind of, summary, he could acquire some new motor skills. He could even, and this is a very surprising thing that made a lot of debate, he could draw the floor plan of his house, even though he moved to the house five years after the surgery. So, and we'll see in a minute, this is kind of surprising because many people believe that the role of the hippocampus is actually to have a special navigation map, and he doesn't have a hippocampus. How that can happen? So the explanation is, if you want to believe it, that he has something to do with motor skills, that he moves into the house, he can somehow, I don't know. And he had an intact working memory. So the idea is that he can talk to you and have a relevant conversation, but he doesn't remember afterwards that he had it. But the working memory is like if you give him a task to remember a telephone number, he can do that if while he rehearsing the number, and he can do that for a while, but nothing will actually keep in memory. So one of the stories I heard, not first-hand but second-hand, from a guy that was working with him, is that you could take him to lunch. He would eat the lunch. The waiter will take the lunch, everything dishes out, and then come back, ask him if he wants to order something, and he would order another lunch. And he completely forgot that he ate, and only after like the third time, when he's kind of completely full, he felt that he's kind of full and he's not ordering it. So it's kind of very weird phenomena, and it's for reasons that I think for me hard to appreciate, because this happened in 50s, 60s. This was revolutionary in some sense in terms of memory, because before that, psychologists and cognitive scientists were trying to untangle these kind of things of memory, this kind of very vague thing, and they try to separate it into different things. And this was the first time that it was kind of shown that you can base some of these distinctions on functions of specific location in the brain. I can tell you personally, this doesn't speak to me. I just cannot attach to that. It's just kind of too strange. So if you like, you can remember this map. If you believe it, good. If you don't, nothing happened. Okay, it is important to realize that the memory is a process. It's not like a computer memory in a sense, and it kind of has a time course, and people kind of again, this is in a way we're trying to impose on that all kinds of things. So people usually split it into acquisition, into consolidation. So acquisition is like when you acquire the memory, new information comes in, and you kind of store it, and then you kind of store it, transfer this information into some kind of a long-term version, and then you store it, and then at some point you retrieve it, but you can retrieve it anywhere during this process, and ask yourself, what happened, and this degradation with time. If you wait, it will go away. Some of them, and one thing to remember is it is important is that this there was this term which was coined, I don't remember by whom, but I will show you in a second, which is called engram, which is talking about the process during which during consolidation, something has to change in the brain. It is like a physical change that keeps the trace of this information. Okay, so here this was this guy Ribot, and he was the one coined the terms engram, a memory trace in the form of physical change in the participation of neurons. Okay, neuron synapses, whatever. So for me, a very good analogy for that is basically, I don't know how many of you, but all of you should know how that works in a computer, right? And in the computer, what we have is, for at least in the current one, what you have is a random access memory, which is basically what holds the information. But if you take the plug out, it will go away. Okay, so all the memory that is in the computer, when you open, you're writing you're too young to know that, but you're writing a document, and there's a power cutoff, and everything you wrote is gone. Okay, this today doesn't happen because the computer is saving it to disk very, very often. But 10 years ago, this would be the major reason for suicide in the West. So this is random access memory, and the reason is that the physical memory is kept on a circuit, on an electrical circuit that needs to keep refreshing. So you kind of you put them to some energetic state, and then they decay. And if you don't do that like hundreds of million times a second, it will just go away. And this is why you need the power to keep feeding the thing off. But when you press the save button, what happens is that this information is copied to a different system, like the hard disk, or in today's case, flash drives. And there, the energy state is kind of stable. It's a stable state. You put the electrons in, they don't go away. They're trapped there, and they keep there. Or in a hard disk, you kind of change the magnet. You use a magnet to change the magnetism of a specific point on the disk. And this could kind of stay there for a long time unless you put it into, you put a magnet on it. So this is in a way the way people envision what happens in the brain during memory. And personally, I must tell you, I'm very interested in the first part, which is, I think, for me, is the biggest question: how does the brain actually save the information before you cut the power off? Okay, what happens if you stop every protein synthesis in the brain? How can you actually keep the memory going? And we do, and we represent the information in terms of like spike trains that traveling across the brain, and this is something that we really don't understand. It's complete mystery. Okay, the fact that you can synthesize proteins and then keep the memory for at least a few hours or days, that makes sense, right? We know this is biology. The fact that the brain is a biological system that actually able to keep the memory ongoing without the biological machinery, that's what I think makes it fascinating for me. But anyway, the term is engram. This is what people are looking for. What is actually changing in the brain during this consolidation process? I want to also mention that there are different types of acquisition of memory. Some of them are incremental, so you kind of train, you play the piano every day, you're getting better, something is changing all the time, okay? But you have to do it, okay? Some of them are single trials. These are the the favorites of neuroscientists. You put a rat, you put it in the cage, we'll talk about it in a second, you give it electric shock, that's it. It remembers that cage, it will never want to go inside this cage again. Single trial and everything is remembered. And the other interesting part, which is the insight part. So you don't understand anything, and then suddenly you understand it. This is kind of the switch that is extremely interesting in terms of psychology, but also interesting in terms of neuroscience, except that we don't know a good protocol to do that yet. But it's obviously very interesting. It's kind of a phase transition in a sense, that is different from that one. Interestingly enough, not every condition works. So, for example, in classical conditioning with Pavlovian dog, if you put the conditional stimulus and you have some overlap, it will work. If you have like immediate repeat, it will work. If they are together, it will work. But if the unconditional stimulus will precede the conditional stimulus, it wouldn't work. So this is interesting. But here we're going into to this classical. So people working on memory, working a lot on this classical conditioning, especially on this protocol called field conditioning. Anyone don't know what that is? Everybody knows. Okay, so we can skip it. The idea, and then I thought it would be interesting to show you here this famous paper by Inban. So the question is, sorry? In Bar Goshen. Okay? So here's the idea. Okay, you take a mouse. This is a rat, but you take a mouse and you do a field conditioning protocol. Okay? And assuming that the consolidation, I'm not going to talk about the whole paper. I'm considering kind of giving it to you as one of the paper to present. But the idea is that if the memory is stored in the hippocampus, and we learned one thing about the hippocampus is that the output of the hippocampus goes through which neurons? CA1 pyramidal neurons, right? So what will happen if you block the outputs of CA1 pyramidal neurons during the protocol? Okay, so this is what's being done here. What you see here is like you inject a virus and you transfect the CA1 region of the hippocampus. Okay, and you transfect it with a virus that has what we call the halorhodopsin. So halorhodopsin is this optogenetic tool to hyperpolarize a neuron when you activate it with a yellow light. Okay, and you put it on a CaMKII 2 promoter, which means that it's going to transfect pyramidal neurons. So you don't transfect all these inhibitory neurons that we said that are all around this CA1 region. Okay, so and now you put an electrode in CA1, neurons are spiking, you put the light on, they kind of stop spiking. You leave the light, they start spiking again. Okay, so basically the idea here is that whenever you have the light on, you block the activity of CA1 pyramidal neurons. Okay, so there are like, I don't know, 12 pictures in this, figures in this paper with multiple panels. I'm going to give you only two. You take the brain, you transfect CA1, you put two optic fibers in the brain, okay, and you train the mouse with the light on. So basically you put it in the cage, you give the electric shock together with the sound, okay, but you also have the light. So you block CA1 pyramidal neurons, then you test, you put it in the cage with the sound. Does the mouse stop moving? Because you know when they have this electric shock, when they know the electric shock is coming, they freeze. They don't freeze. Okay, sorry, control freeze. The mouse with a halorhodopsin do not freeze. Okay, then you train them again, but without the light, and then you test them again, and they freeze. Okay, so basically. Sorry, and even more interesting, you test them afterwards with the light on. Okay, this is a different issue in a sense, but they stops freezing as well. Okay, so basically you gauge the output of the hippocampus, and it's as if you forgot what you've been through. Okay, with me? Okay. Now another version, which is that's why I wanted to tell you this term engram. It's a paper from last year, and this is very sophisticated in terms of molecular biology. But this is what they do here. Okay, so there are these things called immediately early genes. Everybody heard of them? CFOS. You heard of? No? So CFOS is one of these immediately early genes. And the idea is that when a neuron is spiking a lot, the neuron will express this gene. Okay? So if you would put a channelrhodopsin virus, a virus with channelrhodopsin with a CFOS promoter, it will mean that only a neuron that's spiking a lot will express channelrhodopsin. Okay? So now you do that. This is very sophisticated. Very sophisticated molecular biology. But what you do here, what you want is C-force dependent channel rhodopsin. So you want channel rhodopsin to be expressed only in neurons that were spiking a lot. Okay? But you break it into into an intermediate step, which is called TTA-TRE, and in order for this step to work, so basically C-force so neurons that will be active will express this thing. This thing will in turn activate this promoter, and then you will get C-force channel rhodopsin expression. But this link between the TTA and the TRE is dependent on the drug that you have to inject to the animal, which is called doxycycline. So long that there is doxycycline, so the drug is injected to the animal or given by water, basically this line is broken. So C-force could express, but channel rhodopsin will not be expressed. You take the drug off, C-force is expressed, channel rhodopsin is expressed. Okay, so conceptually that's what you need to know. With the drug in, channel rhodopsin is not dependent on C-force. It's actually not expressed at all. When the drug is off, channel rhodopsin is expressed whenever C-force is expressed. What's the idea? The idea is now we inject these viruses into the dental gyrus, and you see that it's actually expressing in the dental gyrus. And you do the following experiment. Okay, you put DOX on, so this link is off, no channel rhodopsin is expressed, and you get the animal habituated to light inside a specific box. We call it box A. Okay, then you take the drug off, you put the animal in box B, a different box, and you give it an electric shock. Okay, and then you put the drug on again, and you put it in box A, the box that it knows not to be afraid of, okay, but with light. So what's the idea? The idea is the following. We put it in A, it knows this box, and it's not afraid of this box. And it's also not afraid of the light, because it knows nothing happened. Okay? Now we put it in box B, and we activate the electric shock. So what happens? Some neurons in the brain, we expect some neurons in the brain to be activated in order to how do you say, to encode this memory. Okay? But these neurons that are being activated now expressing channel rhodopsin, because they don't have the drug, and then C-force is activated because they are activated. C-force is triggering channel rhodopsin. We have now neurons, the same neurons that were activating during the fear conditioning are now expressing channel rhodopsin. Now we put it back into the box of A, right? And we're activating the neurons. We don't activate the fear conditioning, or not the sound, nothing, just activating the neurons. What do you think will happen? Right? If these neurons are encoding the memory of being, that this is something, that we should be afraid of is happening, if we're activating these neurons, we should be afraid. Okay, so that's the idea. Okay, and indeed, what happens is that just to show you that if you have doxycycline on, neurons are not expressing channel rhodopsin. If you put them doxycycline off in the cage, but you don't do any fear conditioning, very few of them is active. But if you put doxycycline off and do fear conditioning, you have these neurons activated or kind of expressing channel rhodopsin. So these are the neurons that kind of maintain the memory of the fear, if you like. Or actually, the way we want to think of it is as the memory that this box is a box that I need to fear from. The fear is actually encoded in somewhere in the amygdala, but we don't talk about that. And then you see that if you take the mouse and you activate the light, it actually freezes. Okay, it freezes 15% more than a normal mouse in that case. Okay, so basically this is like almost science fiction. This is the closest that you can get in order to implant memories artificially in the brain. Okay? 35 years from now, that's a matrix for you, right?
I'm a bit confused because your interpretation is that this is the memory trace, and I could suggest this is just a pathway, that if I've lost, I won't see any pure expression.
But the difference between that and what we've seen in this paper, where you really block, here you activate it. It's not that you block it, you activate the neuron.
Yeah, it's a pathway.
You activate, yeah, exactly.
But it doesn't mean it's a memory trace. I might suggest that in any fear-induced condition, I will see the same neurons lighting up.
Yeah, so you're encouraged to read the papers, all the controls that you can think of, and more than you cannot think of, and we can discuss it if it's kind of convincing yes or no. But I think the concept is like you taking neurons that were activating during one context that you shouldn't be afraid of. That you should be afraid of, and activate them in a different context.
You don't know that.
What do you mean?
That these are the same neurons.
Of course I know that. I made it the same neurons.
Yeah, but you know that these are different mice.
No, it's the same mouse.
You don't slice them out when in.
Okay, we can talk about it. We don't have time. This is clean. The cleanest, almost as clean as you can go.
Does this imply that the pattern of activity doesn't matter, just which neurons fire?
So it's a good question. I would say that it doesn't, but also, I mean, first of all, note that it's 15%. Okay, so it's not very strong effect. Okay, and secondly, you probably don't use all the neurons. It's leaky. So there are neurons that were activated that shouldn't have been, and there are neurons that are not activated that should be. So it's not 100% clean. And it's a very good question whether, if you would activate it in a different way, you would uncover a different mechanism or not. But the fact that it works, in a way, pretty strongly suggests that activating a population in a pretty crude manner is good enough for getting pretty crude results. Okay? Okay. Okay, last topic, because we don't have time. So spatial navigation, we can't do without that. Tetrads. Anyone knows what tetrads are? Tetrads is you take like a very, very thin wire, you take 4 of them and you twist them and then you chop. And what you have is like something that looks like an electrode, but you have like 4 points here that can record voltage extracellularly. There are better ways to do that today. You can kind of put it on a silicone, and then you can have like 4 contacts here. And the idea of these 4 contacts is that if you have the tetrads within a population of neurons, it's like having like 4 antennas, and you can kind of triangulate where the neurons are. So one neuron will give you one signature, so it will be big on one wire and small on other wires, and another neuron will be big on one wire and smaller on other wires. And sometimes you'll have 2 neurons that looks exactly the same on channel 3, but because you have the others, you can actually separate them. Okay, so that's the idea. You can put now hundreds of them on these kind of devices that looks like that, and then implant it on the head of an animal, and then the animal can run with his wires, and you can record the activity in the brain. Okay, this been done first in 1972 by, or maybe not the first time, but this was first people to use it for this task, O'Keefe and Nadell. And what they showed is that when they put an animal in the arena, this is kind of new in the sense that you can have a freely moving animal in space and record activity of the brain. It was very difficult to do before. Note that we only record spikes. We don't know what happens inside the neurons, but okay. Anyway, some people think it's not important. And you see that the idea is that you have like different neurons, and you see that some neurons are like to spike in area C, some neurons like to spike when the animal is moving from A to B, okay, and things like that. So the idea is that you have neurons that are selective for the place in the space. Okay, this became like a major field of neuroscience. Everybody was doing it. You could map, a whole area. This cell is spiking here, this cell is spiking here, this cell is spiking here. The name is place cells, and this is obviously, I wouldn't talk about it if they were not in the hippocampus. Okay, so these are neurons in the hippocampus. Most of them CA1, but also some CA3, also in CA3. So the idea is that these neurons are encoding space. Okay, and then you can do, I don't know how many, 400 PhDs probably were done on that. You put a mouse, you put it in an arena. This is a place cells. You kind of rotate the walls, okay, and the place cells rotate because they're kind of somehow relative to what's going on in space. Okay, you rotate it to the other direction, it rotates in a different direction. Okay, so it's encoding the space relative to whatever. Otherwise, there's no meaning to that. They don't have GPS, right? So they kind of have to rely on something. Now you take the arena, you make it bigger, and it scales with the arena. There is a property called remapping. So you have a cell. So this is a better example. You have a cell that likes a corner. Okay, you put the animal in a different arena. The same cell is now encoding a different place of the arena. You bring it back to that arena. It kind of encodes similarly to that place, and it's like the argument whether it's, if it's exactly the same arena, whether it will kind of remap every time. So if you wouldn't do that in the middle, then it probably would keep that thing exactly here, but because you kind of put it in that arena, it will move. So this is another phenomenon, pretty complicated, this remapping thing. So what I wanted to show you is just, because we really have like 2 minutes, we don't even have that. Few new things that happened since 73. So recently, people started doing that. Anyone heard about this virtual reality? All of you? Doesn't seem like that. Okay, so here's the idea. This is like from, I don't know, 2009. It's pretty old. You have a ball made out of styrofoam, so it's like a calcar, very very lightweight. Okay, you put it in some kind of a bath, and you have airflow going from below. So the ball is floating in the air, but it doesn't move. So it can move, like rotate around itself, every direction. You put a mouse and you hold it in the head on the ball, and then the mouse can walk on the ball. Now you have some optical devices, so it's like a mouse here and then a different kind of mouse here that kind of reads that the ball is moving, okay, and you feed back that into a computer. So when the mouse is going to the right, the computer knows that it goes to the right, and it has a projector like that projects it on a screen, okay? And so when the mouse is running to the right, you can make it like a virtual reality that the environment will move to the right. So the mouse is running in a maze, but it actually keeps staying at the same location. So this is a movie that shows you what it looks like. The mouse is running on the ball. It runs in the corridor. You see that it actually runs on the right side of the corridor, like it would do on a real maze. And if it goes beyond the wall, it will get an air puff, so it would go back. Okay, where it gets to this green point, it gets water. So he learns actually to run to the water, actually, and to get the water, and then he turns around and goes back. Okay, so it's actually a linear track, but in this case you don't see it, but here on the left you can see that they make the walls different. So when it goes through, there are different textures on the side, so it's like creating a maze that changes with time. Okay, and what you can see is that it runs back and forth, back and forth, back and forth, take the water. At the beginning it's hard, and after 10 sessions you do it very, very quickly. And you can find place cells. So the idea is that the mouse is now held in the same position. you can record intracellularly from the brain, from a single neuron, and you can see that this neuron is firing more in one location, another location, so it has place field. Okay? Similarly, you can do the same thing under a 2-photon microscope, and you can image a population of neurons while that's happening. So if you want, again, you have like a mouse running in. So what you see here is it's running in this maze, which is linear track, but it changes the texture, and it takes some water, and then it runs here, and different neurons activate at the different times, and you can actually map that and have receptive fields. So you can kind of record from many neurons, day after day, from the same neurons, and see kind of what they're coding for. And lastly, you can even, this is like, when was that published? I think this year, the beginning of this year. This is by Yaniv Ziv, who is now opening his lab in Weizmann. So what they were doing, they were implanting a camera that is so small that you can actually hold on the mouth of the brain and connect it with a USB cable, and you make basically a hole in the cortex, so you can go all the way to hippocampus, CA1 pyramidal neurons, and I told you already that they are kind of pointing downwards, so you can see the soma, right? And so this is what you see. So you can see hundreds of neurons, okay? And you can record calcium from these neurons, and you can actually map receptive fields. So what you see here is like it's an arena, and the mouse is running in the arena, and there's a different arena, and these neurons spike here, and you have a place field here, and different neuron, it spikes here, and you have a different here, and you can actually do this remapping and go like day after day and see whether this neuron changes its activity, yes or no, and they kind of see that like neurons kind of like the same place, but they're not always activated in the arena. So they can kind of, on one day they would be, neuron 1 would be activated, on day 2 it wouldn't, and day 3 not, but on day 10 it will be activated at the same place that it was activated on day 1. So there's a stability of this receptive field. So this is what it looks like. This is the activity of the neurons, and this is the mouse running in the arena with its USB cable attached to the computer. Okay, so you've been to this talk by Yitzhak Fried, and some of these place fields are also exist in humans, in the hippocampus. So this is just a slide to finish. I think that's it. That's what I wanted to tell you. Questions?
Could HM navigate?
Could HM navigate? Could navigate in his own house. I don't know more than that. I guess no. In a way, I guess not, but I don't know. Yeah, that's a but it's not that he remembered the plans of the house. He could draw it, which means that to some extent he could kind of say, you know, I'm walking this way, I'm going to the kitchen, I'm going back. So in his own house, he could navigate. Yeah, I wouldn't build. I think it's like almost useless to go into this discussion about what HM could do and not do. That's my personal opinion. It's interesting as an anecdote, but it's like, you know, this Oliver Sacks stories about the man who thought his wife is a hat, right? It's kind of it's interesting, but.
What happens to the face cells when mice are not moving at all, but they're showing movies of moving through space? Is there something?
I don't remember. So what you want to say is like, is it like it has to do something with the actions that them exploring themselves?
That they say, I want to go into this direction because something is going on there, or if it's also happening, if they're not moving at all, they're just seeing and remembering something's going on there, but it's not
It's an interesting question. I don't know. So it's like as if you put the mouse and driving it on a car through space. I don't know. I can tell you that, from personal experience, people who sit by the driver don't remember the road, but the driver do. And maybe HM wouldn't. It's a very good experiment to do, I think, especially with these tools. It's kind of almost obvious. It's kind of very easy to do. It would be interesting to see if it actually activates and if it activates the same cells. Good question. I don't remember seeing that, but it's kind of almost obvious, you're right.
Are you going to mention the reactivation of plate cells? Because maybe it's interesting in the context of this question that when the mouse runs on a linear track with several plate cells being active, then arrives at a goal destination and rests, that then the same plate cells are being reactivated but 20 times faster. So this is some kind of...
Yeah, we haven't gone to all of that. There are all kinds of, and they are like very, very new about the fact that it's like these are kind of predicting. There are also neurons that are predicting in the hippocampus whether you're going to, so you can do a maze, a T-maze, and the mouse is going, and at the end of the maze he would turn right or left in terms of like getting the reward here or there, depending on something that's happening here, and you can see neurons that will encode his decision even before he's going there, and would kind of fire the direction that he's going to go through. That very interesting thing. I still have to choose the best papers to go through in our discussion. There's also this subject that I haven't gone through, which is a replay. It's a big thing in hippocampus. We haven't had the time to talk about it, but there are evidence that the sequence of activation of neurons in the hippocampus is replayed after you kind of do that. So, for example, you can, when the animal sleeps, so you take the animal through a maze. I think this is what you're kind of talking about. So you take an animal through a maze, and neurons are active at different places through the maze depending on the path that the animal has taken. All right, and then animal goes to sleep. You continue to record from the same neurons, and you see the same pattern of activation. Neurons are active, one after the other, more or less in the same pattern that they were doing during the day, only faster. And kind of people kind of claim that this is like hippocampus. The idea was hippocampus is replaying the memory to cortex, and then the cortex kind of learns it, and this is kind of the stage of consolidation. Obviously, there are a lot of arguments about it because people were doing recording in cortex and hippocampus at the same time, and shown that actually the cortex was what is called downstate, so it's kind of inactive when the patterns are replayed, and actually, so it's kind of is telling the cortex what it needs to remember, but the cortex is asleep and not listening. So it's kind of exactly. So it depends of but it's kind of random. That's the thing. It's kind of anyway big argument again, but it's a very interesting phenomenon, of course, that this replay thing. And I thought this is another paper that we might want to go through. Big hippocampus. Okay.